Good morning, everyone. Affectionately yours, I'm Dr. Yasmin Kazi, respectfully presenting at this Tuesday CME today on the topic of designer babies. Is it the best thing to happen to us, or is it an ethical horror waiting to happen? The story. Comfortably seated in the fertility clinic with Vivaldi playing softly in the background, their little son, you and your partner are brought coffee and a folder. Inside the folder is an embryo menu. Each embryo has a description, something like this. Embryo number 78, male, no serious early onset disease, but a carrier for phenylketonuria, which is a metabolic function that may cause behavioral and mental disorders. Carriers just have one copy of the gene, so don't get the condition themselves. There's a higher than average risk of type 2 diabetes and colon cancer, lower than average risk of asthma and autism, dark eyes, light brown hair, and a male pattern baldness. A 40% chance of coming in the top half in SAT tests. There are 200 of these embryos to choose from, all made by IVF, from you and your partner's eggs and sperm. So over to you, which one will you choose? Designer babies. Babies who are designed through a genetic modification process are called designer babies or genetically engineered babies. Genes play an important role in our life. If the genes of the embryo are altered using technology, adding in desired characteristics and taking away the undesired ones, then the resulting embryo will have a genetic makeup that has been engineered through gene therapy. Now, what is a designer baby? A baby whose genetic makeup has been artificially selected by genetic engineering to ensure the presence or absence of particular genes or characteristics. In October 2000, Adam Nash was the world's first designer baby born by PGD, that is, pre-implantation genetic diagnosis. PGD refers to screening procedures performed on the embryo to check for genetic disorders prior to implantation and pregnancy. Combined with IVF, a process by which egg cells are fertilized by sperm outside the womb. Embryos grown to the 8-cell stage are checked for genetic disorders. It's a major treatment in infertility, but is now being used for other reasons. To date, genetic technology has only been used to treat serious disease in children. Discoveries about the influence of genes on human traits opens the possibility of transferring these techniques to human beings. Developing technology makes it possible to alter anything from gender to disease and eventually appearance, personality, and IQ. Some believe that parents will inevitably want to choose their children's genes, thus creating designer babies. So I could want to have a baby like this, or couples in future could want to have a baby like this, who looks like this, because it's their choice. Now, types of designing babies. Germline engineering. Germline genetic modification is a form of genetic engineering which involves changing the genes in the eggs, sperm, or very early embryos. This of engineering is inheritable, meaning that the modified genes would appear not only in any children that resulted from the procedure, but in all succeeding generations. Another way is PGD. It is the genetic profiling of the embryos prior to implantation and sometimes even oocytes prior to fertilization. PGD is considered in a similar fashion to prenatal diagnosis. Since 2004, we know that the whole human DNA by the Human Genome Project. With increasing computer power, it was possible to read all the base pairs, which was about 3.2 billion for one DNA copy, which are the basic modules in the blueprint of life. This was developed first in 2012, which uses natural enzymes to target and snip genes with pinpoint accuracy. This method is known as the CRISPR-Cas9. It seems likely that gene therapies eliminating mutant genes that cause some severe, mostly very rare disease might finally bear fruit if they can be shown to be safe for human use. Clinical trials of this CRISPR-Cas9 technology is still underway. Now, what is this technology? It is a clustered, regularly interspaced short palindromic repeats. A map is used to find the exact place where it is supposed to cut in the DNA sequence. After having localized the region on the DNA, the defective part is snipped out, replaced by cutting it out. That's where this Cas9 comes into play. Cas9 is like a pair of scissors. It's a DNA scissors that cuts, dissolves the connection between the elements of the DNA backbone. So you can see the DNA sequence where the CRISPR is working, selecting the area which has to be cut, and the Cas9 goes and cuts it. After Cas9 has successfully found and cut the target region, the defective part of the DNA needs to be changed. For this step, scientists take an advantage of the body's repair mechanism called the homology-directed repair, the HDR. Each cell uses the HDR mechanism to repair and break its DNA. HDR uses a protein complex which is similar to the Cas9. This protein complex is a kind of glue. However, it is not only gluing together the free ends of the DNA strands, but also replaces the adjacent area in the DNA based on the intact template of the whole region. The reason for this is that the area around the breakage could also be damaged, and thus it's safer to replace it at the same time. Scientists produce a lot of DNA strand fragments that match exactly the area around the breakage. But some parts are different, containing new or altered information. They sneak a lot of these manipulated DNA fragments into the cell. When the HDR looks for a template to fix the cut in the DNA strand, it will most certainly take one of these matching but modified fragments the scientists snuck in because the area around the breakage is surrounded by them. But what about modified babies? CRISPR-Cas9 has already been used to genetically modify human embryos in China to see if it is possible in principle. The results were mixed. And Kathy Niakan of the Francis Crick Institute in the UK has even been granted a license by the HFEA, that is the Human Fertilization and Embryology Authority, to use this on embryos a few days old to find out more about problems in these early stages of development that can lead to miscarriage and other reproductive problems. So definitely this procedure is in. Most countries have not yet legislated on genetic modification in human reproduction, but all of those that have all banned it. The idea of using this CRISPR-Cas9 for human reproduction is largely rejected in principle by the medical research community. Now, what are the advantages? Everything cannot be bad. This allows couples who can't conceive normally to have children. It allows couples to balance the gender in their families. Genetic screening reduces the baby's chances of being born with a serious genetic disease. It increases the likelihood of a healthy baby, reduces the chance of miscarriage, reduces the chances of termination due to disorder, and can be used to save lives. Now, what are the disadvantages? Moral and ethical concerns. Is too much playing like God. Social concerns. There is a high cost gap. It leads to gap in society. So people who are rich can afford to have designer babies, and people who are not will remain poor. So the designer babies will then rule over the poor. Genetically enhanced people may start to feel superior to those who haven't been enhanced. Such groups of people may become prejudiced against one another due to a feeling of lost common humanity with non-enhanced people. This cannot lead to ovarian hyperstimulation during the IVF procedure. Unskilled lab technology could damage embryo during biopsy, and God knows what can happen from that. The removal of cells from 8-cell embryos has implications for the well-being of people created by PGD. So these are the samples of design. And this lady says, we would like to have a blonde-haired, blue-eyed girl when they have black. So the survey was conducted in the US about the uptake of genetic engineering and the formation of designer babies, which are being actually offered by some clinics. The survey results showed 56% supported using genetic tests to counter blindness, 75% supported using genetic tests for mental retardation, 10% of the respondents said they would want genetic testing for athletic ability, 10% for improved height, and 13% backed the approach to select for superior intelligence. Now, how is a designer baby created? We just told you about the technology. An embryo is created by IVF. A single cell is removed from the embryo within the first 5 days of its creation. The cell is genetically tested. The parents then decide whether to discard the embryo or implant it in the mother's womb. Single gene disorders caused by the alteration or mutation of a specific gene in the affected person's DNA. Single gene disorders are heritable and often run in families. Chromosome abnormalities usually happen as a result of error in cell division. Multifactorial disorders is caused by interaction of genetic and sometimes also non-genetic environmental factors. So these are the genetic disorders looked out for during the creation of these designer babies. So single gene disorders, chromosome abnormalities, and multifactorial disorders. Genetic counseling can help prospective parents who have a genetic disorder regarding to their future children, because genetic counsel is the counseling of individuals and of prospective parents regarding their offsprings on the probabilities, dangers, diagnosis, and treatment of inherited disease. If allowed, what is the danger? It can have a bad impact on our world. It will end many people thinking that foreigners and anyone else who seems are disgusting and people are scared of them. People who have genetic defects would be socially excluded from other kids. They will be separated from the society. Designer babies will lead to injustice on the basis of certain qualities or traits. Kids of rich families will get genetic enhancement, which I already said, which will lead to genetic upper class people. Parents who love sports would have the athletic ability engineered into the child. However, the child may not want the same. If a child is born with the outstanding ability to play athletics and they choose not to, an engineered gene is wasted, or the child is forced to do something they don't want to. Another reason for making a designer baby is risky are the several mutation, virus, and diseases that alter the genes. Nowadays, it's difficult to know when altering genes whether it can have birth defects and illnesses that will affect a child's life forever. Even though most parts of the world are still male-dominated, the gender of the baby can lead to gender discrimination all over the globe. In some cases, designer babies can be a good thing. For example, the Hashmi family have a child who have a rare blood disorder who urgently needs a bone marrow transplant. Now, the Hashmis may be able to have a child that will be free from the disorder than the other child has. The child that is going to be born could also donate tissue like cord blood to cure its sibling. This is a positive side to designer babies. In many cases, when a child is suffering from disease like leukemia, a parent might have another child, but alter the genes to make it a perfect match to their sick child in order to save the first. Now we go to an interesting case study where genetic engineering and a designer baby has actually been born in the world. Recently, Gyne, a Chinese scientist, claimed to have created the first gene-edited babies designed to be naturally immune to the HIV virus. The news immediately triggered widespread criticism, denouncement, and debate over the scientific and ethical legitimacy of his genetics experiments. China's guidelines and regulations have banned germline genome editing on human embryos for clinical use because of scientific and ethical concerns in accordance with the international agreement. His human experimentation has not only violated these Chinese regulations, but also breached our ethical and regulatory norms. Two beautiful little Chinese girls named Lulu and Nana. This was the first gene-edited babies amid denial from hospital and international outcry. But he did his presentation in one of these conferences where he claimed that he did it and he has created babies who don't have the gene which can cause HIV. A lot of international outcry, a lot of articles, a lot of newspaper articles, and there was a furor. Now, what is the furor about? These include questionable scientific value, unreasonable risk-benefit ratio, illegitimate ethics review, invalid informed consent, and regulatory misconduct. This series of ethical failings of he and his team reveal the institutional failure of the current ethics government system, which largely depends on scientists' self-regulation. The incident highlights the need for urgent improvement of ethics governance at all levels, the enforcement of technical and ethical guidelines, and the establishment of laws relating to such bioethical issues. He announced that he had used the genetic editing technique known as the CRISPR to change the DNA of human embryos created by Ivy. and that these two embryos had been successfully implanted in their womb and developed into these twin baby girls, Lulu and Nana. The experiment has been universally condemned by scientists. In this experiment, He Jiankui used CRISPR to disable a gene called the CCR5 gene in the human embryos. This CCR5 is involved in the immune system, and the HIV virus exploits it in order to infect the human cells. So by disabling this gene, he aimed to make the babies resistant to HIV. But the resistance can be to other things as well, important things. So what is it? Is it good or is it bad? The children won't have HIV. Is it good? Is it bad? Is it money? So what is the future of designer babies? The simplest and the surest way to design a baby is not to construct its genome by pick and mix gene editing, but to produce a huge number of embryos and read their genomes to find out the ones that most closely resembles your desires. Why there is so much international outcry? Because designer babies are like cloning. You can produce batches and batches and batches of baby for organ donation, for human trafficking, and it breaks the entire social structure of the world. Instead of doing that, these are the alternate ways we can resolve it. One is to produce a lot of embryos. Two technological advances are needed for this to happen. The production of embryos for IVF must become easier, more abundant, and less unpleasant. Gene sequencing must be fast and cheap enough to reveal the traits an embryo will have. Put them together and you have something called easy PGD, a cheap and painless way of generating large number of human embryos and then screening their entire genomes for the desired characteristics. To get much broader use of PGD, you need a better way to get eggs. The more eggs you can get, the more attractive the PGD becomes. One possibility is a one-off medical intervention is ovarian tissue freezing. An even more dramatic approach would be to grow the cells from stem cells, the cells from which all other tissue types can be derived. Stem cells are present in the cord blood, which could be harvested at the patient's birth and frozen for later use to grow organs or eggs instead of organ donation. Even mature cells that have advanced beyond the stem cell stage and become tissue-specific types can be returned to a stem cell-like state by treating them with biological molecules called growth factors. Last October, a team in Japan reported that they had made mouse eggs this way from skin cells and fertilized them to create apparently healthy and fertile mouse pups. Thanks to technological advances, the cost of the human whole genome sequencing has also dropped a lot. In 2009, it was around $50,000. Today, it's only $1,500, which is why several private companies can now offer this service. In a few decades, it would cost just a few dollars per genome. Then it becomes feasible to think of PGD for hundreds of embryos at a time. The science for safe and effective easy PGD is likely to exist for some time in the next 20 to 40 years. It will then become common for children to be conceived through IVF using selected genomes. Forecast this, that this will lead to becoming sex being obsolete for procreation. So it seems, concluding my presentation, it seems immoral, unethical to test embryos for certain genes and to tamper with them. Technology is still developing, so safety concerns are high. Many children's lives could be saved. Fewer children will be born with genetic disorders. The idea is still new, so it's hard to say who is right and who is wrong. There are many advantages and disadvantages of a designer baby, and we should keep an open mind and unbiased attitude towards them.